So after the midterm lecture, or after the midterm lab exam, we did an experiment, the effect of UV radiation on bacterial growth. So of course you remember that UV causes damage to the DNA because two adjacent thymines, most often two thymines, are excited by the UV radiation to cross-link, and that creates a thymine dimer. And the problem is that since bacteria replicate and divide in as little as every 20 minutes, you can overwhelm them with so many thymine dimers that they cannot, replicate, that they can't reproduce, so they die. And the problem with the thymine dimer is that when DNA polymerase or RNA polymerase comes along and sees that thymine dimer, it cannot add the correct complementary adenines. And so a mutation occurs. And if there's too many mutations, those cells die. So can everybody see the handout on UV?
Yeah, we can see.
So what we did is we covered the entire surface of the agar plate with our bacteria, and we were using Serratia marcescens, which produces a deep red pigment. And then we exposed two plates to UV without the plastic lid on. So the first plate was exposed for 20 seconds and the second plate for 2 minutes. The third plate was exposed for 2 minutes, but we put the plastic lid on. And the fourth plate was just a control. And we also had little paper masks, right? Paper cutouts, so that the UV would only go through the opening of the cutout. And so what we observed was that even with a 20-second exposure, the UV killed the bacteria on the plate. And so we saw a design from our paper cutout. And with a 2-minute exposure, we had a lot more killing. So increasing exposure time is more effective at killing the bacteria. On the third plate, we did leave the plastic lid on and expose the bacteria to 2 minutes of ultraviolet radiation, and we still got some killing. So enough UV passed through the polystyrene plastic to kill the bacteria. We also noticed that we had some non-pigmented colonies growing in the kill zone, and those non-pigmented colonies were mutants. They had a mutation in the pigment gene, so they could no longer produce that deep red pigment, and that was caused by UV exposure. So we saw that polystyrene plastic blocks some UV, but not enough. It can still penetrate enough to kill, right, to destroy cells. And the paper mask, paper does also block UV. So we saw increasing exposure time is more effective. Paper blocks UV. Polystyrene plastic does not block enough UV. And so what kind of plastic did we say is very effective at blocking UV and is pretty cheap?
Was it polycarbonate?
Right, polycarbonate plastic. So polycarbonate is cheap. It blocks UV. It's not as cheap as polystyrene, but polystyrene is not good for blocking UV. So UV causes thymine dimers specifically, or pyrimidine dimers in general. And the idea is if you have too many of the thymine dimers, you get too many mutations, and that kills the bacteria. We left the lid on to see whether or not polystyrene plastic does anything. It does block some UV, but not enough. And the pyrimidine dimers, like thymine dimers, right, they lead to mutations because the polymerase enzymes cannot add the correct complementary bases. And yes, bacteria do have a way to repair UV damage, just like we do, but they have DNA repair enzymes that cut out the thymine dimers and replace them. The problem is, as I already mentioned, the bacteria are replicating and dividing quickly. So if they accumulate too many thymine dimers, they have too many mutations, and so that kills those cells. So you can overwhelm microorganisms with UV light. Any questions on UV? So next we looked at some fungi. Remember, for the fungi, the idea was just to be able to recognize the type of fungus from the slide. So the first one we looked at was Penicillium. Remember, these have been stained, but the structures we're looking at here are the conidiaphores, which do have a bunch of spores on the tips. And so the conidiaphores are reproductive structures. And what can you tell me about fungal spores and their chromosome number? What can you tell me about the chromosome number for fungal spores? Somebody knows. Are they haploid or diploid?
They're haploid.
Right, fungal spores are always haploid. And in fact, fungi spend most of their lives as haploid organisms, only occasionally getting together and exchanging genes in sexual reproduction. The Aspergillus is very easy to recognize because it kind of looks like a flower. And the conidiophore of Aspergillus is larger than Penicillium, and it forms a ball, kind of like a puff ball. And this is a slice of that puff ball that has been stained. And we can see it's loaded with spores. Anybody remember what was special about Aspergillus? And I mean, special as in bad. What was bad about Aspergillus? Nobody remembers why Aspergillus can be bad? Because they produce aflatoxins, which are carcinogenic. I know everyone can tell me what's good about penicillium.
It makes penicillin.
Yep, it makes penicillin. Rhizopus is one that's very fuzzy and filamentous. It will produce a lot of asexual sporangia, but the side we're looking at here is the zygote. It's the sexual phase for the rhizopus. And so zygotes are pretty big. So we had two opposite mating strands come together and exchange genes, forming a temporary diploid structure of the zygote. But the spores produced from the zygote will be haploid. So in this case, the spores are produced by meiosis. In other cases, the spores are produced from mitosis, right? Because spores are always haploid. When it comes to yeasts, if everything is wonderful, the yeast just bud like crazy, right? A form of unequal cell division and asexual reproduction where the cell divides unequally. But that's if everything is wonderful, lots of food, clean environment. But if the yeast cells get stressed, then they can reproduce sexually and they will exchange genes and form ascospores. And you can see the dark in this image, they look kind of black, but they're usually stained a real dark red color, so they almost look black. And those are the spores inside of the green yeast cells. So when conditions are not favorable, fungi will get together, exchange genes, and produce spores or offspring that hopefully are better adapted to survive. But we said before, if everything is wonderful, then you don't need to enhance variation or add diversity. You can just reproduce asexually. The main point here is to be able to recognize the fungi when you see them. So any questions on the fungi? Next, we talked about protozoa. In particular, we talked about two parasites, trypanosoma and plasmodium. So it's important to be able to recognize these parasites when you see them and to know these characteristics about them. So our first protozoan parasite is plasmodium, which causes malaria. It's transmitted by female mosquitoes. We talked about how they need the blood, that extra nutrition, for their development of their eggs and larvae. It is an intracellular parasite, so it's small and lives inside of red blood cells, also known as erythrocytes. And like all protozoa, there are many stages to the life cycle of Plasmodium, but we're concerned with the signet ring stage. So you can see inside of these red blood cells, we have the signet ring stage, right, for Plasmodium. And depending upon the staining technique used, the colors can be different. But we do see the ring stages. So that's for Plasmodium. When it comes to trypanosoma, this parasite causes sleeping sickness. It's transmitted by a fly that goes by different pronunciations, tsetse, setse, cheche. And this parasite is much larger than Plasmodium. So it's an extracellular parasite. Trypanosoma is found in the bloodstream, but not inside of any cells. You see, it's pretty large. They have a flagellum. You can see the nucleus inside. So those are the two parasites. For the live protozoa, we looked at some videos, and even if we were meeting face-to-face, we would not have any live protozoa slides on the lab exam because that's just too hard to maintain all day long. But it is important to know the types of locomotion, the types of motility that protozoa use. And they can use flagella, cilia, and pseudopods. Which type of motility do bacteria use? What do they use?
What's the question again? Sorry, I was reconnecting.
How do bacteria move? Which of these types of locomotion do bacteria use for motility?
Flagella only?
Right, just flagella. So we did look at some videos of these organisms, like the Euglena, which was green, had one flagellum. We've seen paramecia, which are covered with cilia, and the amoeba crawl around using pseudopods. So you should know the mechanisms of locomotion and give some examples of protozoa. Any questions on the protozoa? Selective and differential media. So we talked about this both in lab and lecture. So remember, a selective medium allows some microorganisms to grow while inhibiting others. A differential medium allows the organism to grow, and as they're growing, they display a characteristic, usually carbohydrate fermentation, which allows us to distinguish one type of microorganism from another. And usually that's with a color change. A medium can be either selective or differential or both. We put chemicals in that allows a medium to do what it is supposed to do, either select or differentiate. So we had MSA, which stands for mannitol salt agar, PEA, phenyl ethyl alcohol agar, MAC, short for McConkey agar, and EMB was eosin methylene blue agar. So the MAC and the EMB, they both do the exact same thing. They both select for gram-negative and inhibit gram-positives, and they both differentiate lactose fermentation. But they do this using different chemicals, so the colors are different. On the EMB, there's really only one color we care about, and that's the shiny green color of E. coli. Because E. coli loves lactose, it ferments lactose so well, it makes so much acid that the eosin and methylene blue stains crystallize, and you get that beautiful shiny metallic green color. For MAC, there's only two colors to worry about here, pink and not pink. So if the bacteria ferment lactose, the growth turns pink. If the bacteria do not ferment lactose, then it's just the ordinary color of the bacterial growth, so it's not pink. For MSA, the mannitol salt agar, MSA is both selective and differential. It's selective because of the high salt. So there's 7.5% sodium chloride. So that's more than twice as salty as the ocean. And that high salt inhibits a wide variety of bacteria. But the only reason why we ever use MSA is to grow Staphylococcus. And the MSA plate also contains mannitol, sugar alcohol, which is fermented like a carbohydrate to produce acid. And so the MSA also contains phenol red, a pH indicator that turns yellow with acid. So we're looking to see if the agar turns yellow on MSA. And since we only ever use it for staph, which species of staph ferments mannitol and turns the agar yellow?
Is it Staph aureus?
Yep, Staph aureus. So we can differentiate Staph aureus from Staph epidermidis or most other types of staph because Staph aureus ferments the mannitol. And the PEA plate, the phenyl ethyl alcohol, inhibits gram-positive bacteria, or inhibits gram-negatives and selects for gram-positives. So it's the only one that's selective only and allows the gram-positives to grow. So in this first plate here, you see that beautiful metallic shiny green color of E. coli. Underneath the shiny green, it's dark purple. And that's what lactose fermenters normally look like on EMB. And then for McConkey, a beautiful magenta pink. So that tells us this organism is also fermenting lactose. On the MSA, here we have an organism like Staph aureus, which is fermenting the mannitol and producing a lot of acid, which is diffusing through the agar and turning the phenol red to yellow. So any questions about selective and differential media? So then we began performing some biochemical tests. And so when we perform a biochemical test, what are we actually looking for?
The enzyme produced.
Right, the enzyme. We know that enzyme is present if we find the end product of that enzymatic reaction. So the first test was catalase. And as you remember, catalase is an enzyme that breaks down hydrogen peroxide to produce water and oxygen gas. So the oxygen we can see as bubbles. And so you mix your bacteria with some hydrogen peroxide on a slide. If it bubbles, you know the bacteria have catalase. And we talked about how aerobic organisms or organisms that like or use oxygen often have catalase, but sometimes they don't. And yet they love oxygen. So they're producing lots of toxic hydrogen peroxide. So how can they survive? How can they break down the hydrogen peroxide if they don't have any catalase? What other enzyme breaks down hydrogen peroxide?
Peroxidase.
Right, peroxidase. So they could have that other enzyme, right? But we can't tell because the end product is water. With the nitrate test, we were looking to see if the bacteria can reduce nitrate into nitrite. And so the enzyme that does that is nitrate reductase. And so we test for the presence of nitrite. We do that by adding two chemicals, alpha-naphthylamine and sulfonilic acid. If nitrite is present, a red color will develop. So if the bacteria can reduce nitrate into nitrite, a red color will have formed. And that is a positive test result. That means the bacteria have nitrate reductase. If there's no red color, then we have to add zinc. So if we add zinc and the tube turns red, that's a negative result because zinc is a reducing agent. Zinc can reduce nitrate into nitrite. So if the nitrates were still there, right, the bacteria had not done anything to them, then the zinc would reduce them to make the nitrite. And since we already added our nitrate A and B chemicals, now the tube turns red. So that would be a negative test result. And then if you added zinc and still no red color, that was actually a positive test result. And it's positive because there's no nitrate, no nitrite. It's all been reduced, usually to a gaseous form like nitrogen or ammonia gas. And so we call that organism a super reducer. It's still considered positive for nitrate reductase because you have to start at the beginning of the process and reduce the nitrates to nitrite. Then there are further enzymes that continue to reduce everything, right, to a gaseous form. So if you add nitrate A and B and the tube turns red, that's positive. If there's no red color and you add zinc and then the tube turns red, that's negative because the zinc did the reducing, not the bacteria. And if you add zinc and there's still no red color, then the organism is a super reducer and is also positive for nitrate reductase. So questions on the nitrate test or catalase test? So then we moved on to some more biochemical tests, starch, lipid, and gelatin hydrolysis. So we know that we can test for starch by using iodine, right? Because starch and iodine form a very dark product. So we take agar plates, which have a lot of starch in them, inoculate them with the bacteria, incubate them, and next class we add iodine to the agar. if the starch is hydrolyzed, we see a clear zone around the growth because all the starch has been digested, has been destroyed. If the agar all turns dark, then that means the starch is still there and not hydrolyzed. So the bacteria on the right, they have the enzyme amylase, which has digested the starch. So there's no starch left to react with the iodine, so we get a clear zone. In fact, the agar turns clear enough to see through. The organism on the left, no amylase. So all the starch is still there. For lipid hydrolysis, the enzyme is lipase. And so our source of fat or lipid is going to be vegetable oil. And so vegetable oil is composed of triglycerides. And triglycerides are made of glycerol and fatty acids. So the lipase enzyme will split the fatty acids off of the glycerol. When you separate those fatty acids, they become acidic. So we can observe a simple pH change. So the type of nutrient medium we use is neutral red fat agar. So neutral red is our pH indicator. It turns red with acid. Our source of fat is vegetable oil. And so we look for red fat droplets. And so on the right, we can see the Staph aureus has eaten the fat, right? It's broken the fat down. And so we get the red fat droplets. On the left, E. coli, no red fat droplets, so E. coli cannot eat fat. So again, the enzyme is lipase. It splits the triglycerides, separating the fatty acids right from the glycerol, and the fatty acids become acidic, lower the pH, and neutral red turns red. With gelatin, gelatin is our source of protein. And of course, you know, proteins are made up of amino acids. And so intact proteins are able to form a gel. Amino acids cannot form a gel. So we're looking to see if the bacteria produce a special protease called gelatinase. And gelatinase will hydrolyze the gelatin and form a solution of amino acids. And so a gelatin forms a solid at room temperature. But the problem is that when we incubate at 37 degrees, the gelatin melts. It has a very low melting point. So everything turns liquid in the incubator. So after incubation, we have to refrigerate our gelatin tubes, cool them down, and see if the liquid goes back to solid. So if the tube goes back to being solid, no hydrolysis. If the tube stays liquid in the fridge, then that means that the protein has been hydrolyzed into amino acids. So if the bacteria have gelatinase, the tube will be liquid in the refrigerator. If it's solid, no hydrolysis. So what do the enzymes amylase, lipase, and gelatinase all have in common, besides being enzymes? What's special about amylase, lipase, and gelatinase? What's special?
Is it that hydrolysis occurs with all of them?
Something else besides that. Nobody remembers what's special about these enzymes? Think about starch, triglycerides, and gelatin. They're all huge molecules, right? They cannot pass through the cell membrane. So what has to happen to them first?
They have to be broken down.
Right, so that means all those enzymes are extracellular enzymes, right? So they're secreted to the external environment where they can break down those large molecules into small pieces. Then the small pieces can be absorbed by the bacteria, right? So starch, fats, and proteins are big, so they must be digested outside the cell first. So all those enzymes are extracellular. Any questions on starch, fat, or gelatin hydrolysis? Okay, next we had a lot of biochemical tests. A lot more, right? So the first one, the carbohydrate fermentation. So we know that bacteria can ferment a wide variety of carbohydrates, right? They may have a variety of operons that allow them to use those different carbohydrates for energy. And so we can use a nutrient broth medium which contains only one type of carbohydrate, whichever one we're testing. And then the broth also contains phenol red, which we know turns yellow with acid. So if the bacteria ferment the carbohydrate, they'll produce acid, and so we can see that with a color change. In addition, some bacteria, when they ferment, produce carbon dioxide gas. So we have a little Durham fermentation tube, our upside down bubble catcher, and we can see in particular in this tube here, there's a gas bubble. So producing acid with or without gas is a characteristic we can use to help identify different bacteria. And we tested glucose, lactose, and mannitol. We learned that E. coli loves all different kinds of carbohydrates. It ferments and produces gas. So E. coli is a carboholic. And then we also saw that Micrococcus luteus does not ferment anything, not even glucose. How's that possible? Why is it that Micrococcus luteus cannot ferment anything?
Is it because Micrococcus luteus is an obligate aerobe and fermentation is anaerobic?
Yep, so Micrococcus luteus is an obligate aerobe, which means it must have oxygen. And we know that fermentation is an anaerobic process. So obligate aerobes cannot perform anaerobic processes. So Micrococcus luteus cannot ferment anything. So then we started with the IMVIC tests. So IMVIC, IMVC. Indole was the first test. So indole is actually the product. We use tryptone broth, which contains tryptophan, an amino acid. And if the bacteria have the enzyme tryptophanase, they can convert tryptophan into indole. And then we have to add Kovacs reagents to the broth, and we look for that red fuchsia layer on top, and that indicates the presence of indole. And so we tested E. coli and Enterobacter aerogenes. And remember, the new name for Enterobacter is Klebsiella aerogenes, but for our purposes, we're going to continue using Enterobacter aerogenes. And the E. coli was positive, right, for indole. It had that red fuchsia layer, and the Enterobacter was negative. So that was the first of the IMVIC tests. So I'm going to jump to the other test, and then we'll come back to SIM. So the MRVP, right, the methyl red and the Voges-Proskauer tests. So methyl red is easy. Methyl red is a pH indicator. And so we're just looking for the production of acids. And so the methyl red stays red with acid. So that's a positive test result. If it's yellow or orange, that's negative. The VP is a more complicated test. For VP, we're looking for the end product, acetylmethylcarbinol or acetoin. And so we have to add two chemicals, right, to the tube, and then give it 30 minutes to develop. Now remember that we only use one MRVP broth tube. We incubate it, we inoculate it, we incubate it, and then in the next class, we divide the broth. And that way we now have two tubes, one for the methyl red test, one for the VP test. But the VP, after you add your two chemicals, the Barrett's A and Barrett's B, you have to let it set for at least 30 minutes for color to develop. And a cloudy red layer at the surface is a positive test result for the VP test. And so we see here, on the left is our positive VP. On the right is our positive methyl red test. Again, E. coli was positive for methyl red, negative for VP, and the Enterobacter was negative for methyl red and positive for VP. The last test for the IMVIC test is citrate. And so we use an agar slant to do this test. And the idea here is that citrate is the only carbon source in the agar. So the bacteria must be able to absorb the citrate to use it to grow. And so they need the enzyme citrate permease. So if the bacteria are able to absorb and use the citrate, they grow well. Then they use the ammonium phosphate that's in the agar and produce an alkaline product. They produce ammonia, which raises the pH. In this case here, our originally green agar slant changes color to blue, right, to indicate that high pH. So it's a beautiful blue color, indicates a positive citrate test. And so E. coli is negative for citrate, and the Enterobacter is positive for citrate. So you do need to know the results of E. coli and Enterobacter for the IMVIC tests. And it was very easy to remember them, right? What were the test results for E. coli and IMVIC?
E. coli was positive.
Well, for IMVIC, for all the tests, what was it? We have 4 tests.
Oh, right. It was positive for the indole.
Yep. Okay, what about the rest? IMVC. E. coli is positive, positive, negative, negative, right? And the Enterobacter,
is negative, negative, positive, positive.
right, so it's the reverse. Negative, negative, positive, positive. So you only need to know the test results of E. coli and Enterobacter for MVIC. Our next biochemical test is urea hydrolysis. So we have little agar deeps of urea agar, so they contain urea. If the bacteria have the enzyme urease, they will convert urea to ammonia, and that raises the pH. Our pH indicator is phenol red, which turns that fuchsia, that hot pink color, so that indicates a high pH. Yellow indicates acid, but that's a negative test result for this test, right? We're looking for high pH, not low. And then orange, or that peachy orange color, is the original color of the urea agar, and it's around pH 7, so it's around neutral. Our last biochemical test was SIM. So remember, SIM is an agar deep. It can perform 3 different tests. The S is for H2S, or hydrogen sulfide gas. The I is for indol. M is for motility. Normally, we only really use this for the H2S. So hydrogen sulfide is a gas. And we have an agar deep. So the agar contains iron. Iron reacts with hydrogen sulfide to make iron sulfide, FeS. And that forms a black precipitate. So if the tube turns black, that's positive for hydrogen sulfide because iron sulfide has been produced in the tube. We can also put Kovacs on top and do an indole test. And we see here 2 positive indole tests. But remember, for indole, the most reliable source is the tryptone broth. And the reason why, if you have an organism that really does need the oxygen or like it a whole lot, it may not grow well enough in the SIM deep to get a positive indole test. So you might get a false negative just because the organism really likes to have a lot more oxygen. And then for motility, some bacteria have flagella, so they can move through the agar. If the bacteria also produce hydrogen sulfide, then it makes motility easy because the black will be all over the tube because the bacteria have moved all throughout the tube. If it's H2S negative, then you have to compare an uninoculated deep with your inoculated tube to see if the inoculated tube is cloudy, if it's turbid, and then you know the bacteria moved. But normally we just mostly use the SIM for the hydrogen sulfide, right, the H2S. So remember the biochemical test chart, which summarizes everything. So you should know substrate, product, enzyme if we care about it, how you perform the test, if you need to add anything to it like Kovacs or Barrett's reagent, and what a positive test looks like. So make sure you review the test chart. Then we talked about streak plates. Unfortunately, we were not able to make any, but we talked about them. And the idea behind a streak plate is to separate mixed bacteria on the surface of an agar plate. It's also analogous to doing a serial dilution on the surface of a plate because you end up with fewer and fewer cells the more you streak. So we start off by getting a loop full of bacteria and streaking the upper portion of a plate. We then sterilize our loop because all we need is 1 loop full of bacteria. So we sterilize our loop, we drag it into the first streak, usually 2 or 3 times, and then stay out of it, spreading those bacteria in zone 2. Then we sterilize the loop again, drag it into zone 2, once or twice to grab some of those cells and then spread them out. So the idea is we are diluting our sample with each streak. So by the time we finish, we have obtained discrete isolated colonies. And so you can actually tell how many different kinds of bacteria you have mixed together, because remember that as bacteria grow, they exhibit different growth characteristics like size, shape, color, texture, so we can see that. You can also use an isolated colony to create a pure culture, because there's only one organism in that colony because it was started by one cell. But students have difficulty with this technique. Sometimes they use 3 loopfuls of bacteria, right? 1 for each streak. Sometimes they forget to sterilize their loop in between each streak. Or sometimes they just don't get the pattern. And just kind of be creative and make up their own patterns. So that does happen. But ideally, you should have nice multiple isolated colonies in zone 3. Or sometimes you might do a 4-streak plate. But at the end, you want to have those isolated colonies. So which of these plates is the good plate?
Three?
Yeah, plate number 3. Right, so we have those nice isolated colonies at the end. The other plates have various problems, like plate number 1 looks like they used just 3 loopfuls of bacteria. Same maybe with plate 2. Plate 4, they just didn't grab enough cells as they were streaking, and same thing with plate 5. They just never got into the very first streak, so they missed the opportunity to grab bacteria and spread them out. But streaking plates is a very common technique. It's an important technique, as I said, for separating mixed bacteria on the surface of an agar plate. It's also analogous to performing a serial dilution, right? Because you have the most cells in zone 1, and then fewer cells in zone 2, and the fewest cells in zone 3. Any questions on the streak plate? So then we did do a serial dilution using a bacteriophage, a bacteria virus, and its host E. coli. And so we used a T-even bacteriophage like T2 or T4, and T-even phages have a lytic life cycle. So the idea is you mix your phage and E. coli together, you spread that mixture on the surface of an agar plates, the E. coli grow and completely cover the entire surface of the plate, and then the viruses reproduce and kill the E. coli, leaving holes in the growth. We call those holes plaques. So plaques are full of virus. And of course, we are going to count the plaques to determine the concentration of phages per milliliter in the original sample. Now, we've talked about why the plaques stay small. So why is it the plaques stay small? Why don't the viruses just spread and wipe out every bacterial cell on that plate? So why don't the viruses spread and kill all the E. coli on that plate?
It's because they're only infecting bacteria in log phase.
Right, so the viruses want young, healthy, metabolically active cells, and those cells are occurring in log phase. And so log phase doesn't last very long under ideal conditions, and the bacteria actually start entering into stationary phase, soon to be followed by death. and it's not the kind of cells the viruses want. And so the plaques form during log phase, and then they stop. They don't continue to grow and spread. Now the serial dilution, remember, it's the part divided by the whole. It's the volume that you put into the tube divided by the total volume in that tube, and that gives you your dilution. But in this experiment, we started out with a phage that was already diluted 10 to the negative 4. And we were going to continue to dilute it even more. So each broth tube, each dilution tube contained 9 milliliters of broth, and we added 1 milliliter of our phage dilution into that broth. So that's a 1 to 10 dilution, 0.1 or 10 to the negative 1. So we repeat the process. So all of our dilutions are 10 to the negative 1 dilutions. And the dilutions are cumulative. So we start off with 10 to the negative 4. We do a 10 to the negative 1 dilution. It becomes 10 to the negative 5. We do a 10 to the negative 1 dilution on this, and it's negative 6. Then negative 7, negative 8. So those are all of our dilutions. Now, in this experiment, we're mixing the phage and E. coli together in soft agar. So we have a little tube with a little bit of a melted soft agar. And the whole purpose is just to mix the phage and E. coli together. Ultimately, we're still just transferring 1 milliliter of our phage dilution onto the surface of the agar plates. So because the volume we're putting on the plate is 1 milliliter, the dilutions on the plates are the same as they are in the tubes, right? There's no difference because we're using 1 ml, right? So it doesn't change anything. If we were using 0.1 milliliter, then that would change our dilutions on the plate. We're using just 1 ml. So the dilutions on the plate are the exact same as in the tubes, right? 10 to the negative 4 through 10 to the negative 8. So we add our phage to the soft agar, then we add our host E. coli to the soft agar, mix them together, pour that mixture onto the surface of the agar plates, swirl the plates to spread it out, let them cool and solidify, and then incubate them. In the next class, we count the plaques. What is the range of plaques that we have to count?
Was it 30 to 300?
Right, 30 to 300, so in between 30 and 300. Remember, above 300 was too numerous to count, or TNTC, and below 300 was below 30 was too few to count, or TFTC. So we're going to count the number of plaques and then multiply by the dilution factor of the plate. Remember, the dilution factor is 1 divided by the exponent, right? So 1 over a negative exponent, it becomes positive. So it's 10 to the 4, 5, 6, 7, 8. In this example, we counted 147 plaques on plate 4. So that's 147 times 10 to the 7. We have to put the number in proper scientific notation, 1.47 times 10 to the 9. So whenever we take a whole number and reduce it, we have to increase the exponent. And that gives us PFUs per milliliter. What's a PFU?
Is it a plaque forming unit?
Yep. And what forms a plaque?
The virus?
Well, one virus particle, right? So it's a concentration of viruses or phages per milliliter in our sample. So any questions on the bacteriophage plaque assay? Okay, next is the gradient plate. So in this experiment, we create a concentration gradient of streptomycin. We do that by using 2 layers of agar. Our first layer is just plain agar, and the second layer contains the streptomycin solution. which has some green food coloring in it, so some green dye so we can just see it. The thick area is the darkest green, so it's got the highest concentration of streptomycin. As we move across the plate, the concentration of streptomycin decreases until we end up with basically 0 on the very edge of the other side of the plate. We spread E. coli on the surface, incubate the plate. In the next class, we observe that we have a lot of growth where there's little to no streptomycin. As we move across the plate, the concentration of streptomycin is increasing, and the amount of E. coli is decreasing, ultimately ending up with just a few colonies in the highest concentration of streptomycin. So these colonies represent mutants. So these are streptomycin-resistant mutants that were already there in that population of E. coli. Why are those mutants already there? Why did we expect to find any resistant mutants? So why do we expect to find any resistant mutant E. coli? Didn't we learn there's always some resistance present, or just due to random chance spontaneous mutations? So in any given population of bacteria, there's a probability of finding at least a few cells which have those mutations, so they're naturally resistant to basically almost any antibiotic, even before they've ever been exposed to it. So we just demonstrated this with a population of E. coli that's never been exposed to any antibiotics, and yet we find some resistant mutants there. What the streptomycin is doing in this experiment is that it is selecting for those naturally occurring mutants. It's not causing them, it just selects for them. But the mutations are already there in a small percentage of cells, and so we just selected for them. Any questions about the gradient plates? Next, we did a couple of disc diffusion experiments. We did it with some antiseptics and disinfectants, as well as antibiotics. But first, the antiseptics and disinfectants, or the germicides. And so antiseptics are primarily used on living tissue, whereas disinfectants are primarily used on inanimate objects and surfaces. Can these chemicals be used interchangeably?
Sometimes.
Right, sometimes. So not always, not never, but sometimes. Things like isopropyl alcohol and hydrogen peroxide, right, they're interchangeable. You can use them on tissues or inanimate objects. And so in this experiment, we took agar plates, we used swabs to completely cover the entire surface with bacteria, and we had 3 types, E. coli, Staph aureus, and Bacillus cereus. And then we took little sterile paper discs and dipped them into our chemicals and applied the disc to the agar plates, and the chemicals diffused through the plates and hopefully inhibited the bacteria, forming zones of inhibition. Now, the chemicals that we used were isopropyl alcohol, 70%, so rubbing alcohol, hydrogen peroxide, betadine, bleach, amphil, which is like Lysol, and Listerine. And so all of those chemicals should have been very effective at killing bacteria, but the results on the agar plates were not very good for many of these. Exceptions being hydrogen peroxide, which is great for everything, right? It kills everything really well. But other things like Listerine had little to no killing, right? So a very small or no zone of inhibition at all. And so the disk diffusion is not a great test for a variety of chemicals. And the reason why is neutralization, dilution, and diffusion. So agar is organic matter. So it's possible for a chemical agent to bind to the agar and be neutralized. Agar is also mostly water. So your chemical agent may be diluted below its effective concentration. And then large molecules have difficulty moving through the agar, right? They don't diffuse well. So because of these three problems, your zone of inhibition may not have been very good at all, even though all these chemicals are very effective at killing microorganisms. So when using disk diffusion, it's not the greatest test for using different chemicals because there are problems with the agar. So, and we just looked at the results and we just sort of scored them relative to each other, large, medium, or small. These are just some examples of how it's done. It's not using all the chemicals that we use. But again, the problem is that because of the agar and the problems of neutralization, dilution, and diffusion, the chemical agents may not appear to be that effective, when in fact they are. So questions on the germicides?
Sorry, are you able to say the question again?
Yeah, so, well, but the question besides, are there any other questions? The idea was that because of the agar problems of neutralization, dilution, and diffusion, your results do not appear as good as they should be, right? All these different chemicals are very effective. They just may not show that effectiveness on the agar plate.
Oh, okay, I see. Thank you.
Any other questions? The other disk diffusion test we did was the Kirby-Bauer antibiotic sensitivity test. And so in this case, we don't have to worry about neutralization, dilution, or diffusion because the antibiotics are standardized. But when it comes to the zone of inhibition, we do have to measure the diameter of the zone of inhibition. And that's because there's some other issues we have to take into account. And the issue has to do with how your body, right, deals with the antibiotic versus how an agar plate does. So what does your body try to do to antibiotics?
Get it out of you?
Right, so your body excretes in urine and feces, and the agar plate cannot excrete. What else does your body do to any chemical you try to put into your body?
It tries to detoxify it.
Right, using what organ?
The liver.
Right, so there's no liver in an agar plate, so all the antibiotic is there. So there may in fact be a zone of inhibition on the agar plate, but it's not big enough to be considered effective in the body, right? Because your body's going to try to excrete and try to detoxify. You also have to take the antibiotic in a high enough concentration because it has to go all throughout your body to encounter the pathogen wherever it might be. So that's why we have to measure the diameter of the zone of inhibition. So here's an example. We use a ruler to measure the bottom of the plates, and we go from edge to edge through the center of the disc in order to measure the diameter. If you don't have a good diameter, you can measure the radius right from the center of the disc to the outer edge and multiply by 2. But we use the metric system, so we want to measure in millimeters. And so all the discs are labeled, right? TE for tetracycline, P for penicillin, S for streptomycin. So again, we cover the entire surface of the agar plate with bacteria, and then we apply the antibiotic discs, incubate, the antibiotic diffuses and inhibits the bacteria, creating the zone of inhibition, and then we measure it. So here, for example, our zone of inhibition is about 25 millimeters, and that's for tetracycline. So you go back and look at the table. Right, for tetracycline, and we have 25, so that's 19 or more, so that means the organism is susceptible. If you had a small zone, and we don't really have any, on these images here, but if you had something that was really small, and for streptomycin, if, for example, the zone of inhibition, was less than 15, well, it depends on how much less than 15, right? Because 15 or greater means the organism is susceptible with streptomycin, but if it's less than 15, it could be intermediate, so it's beginning to become resistant. If it's less than 11, right, millimeters, then the organism would be resistant. And for the penicillin, which has a huge zone of inhibition, so you can kind of guess that without even measuring, but it's over 40 millimeters. And so for penicillin, whether it's staph or anything else, the organism is going to be susceptible. But you do have to measure the diameter of the zone of inhibition and look at the table. So I haven't seen the lab exam yet, but there may be a picture like what I showed you here, and then you have to measure and look at the table and determine if the organism is susceptible or resistant. So questions on the Kirby-Bauer technique? So once again, how is your body different from the agar plate?
It has ways of detoxifying and excreting antibiotics, but the plate can't do that.
Right, so because your body will excrete the antibiotic and detoxify it with the liver, we have to make sure the zone of inhibition is sufficiently large enough on the agar plate to be considered effective in the body. So then we started doing some experiments on normal flora, right, the normal microbiota. Our first experiment was a Snyder agar. We were testing for the presence of two groups of bacteria, Streptococcus and Lactobacillus, which produce acids. So if you have a lot of those bacteria on your teeth, the acid will destroy the tooth enamel and create a cavity, or technically a carie. So the Snyder agar is both selective and differential. It's selective because it does have an acidic pH around 4.8, so that does inhibit some bacteria, but not Streptococcus or Lactobacillus because they produce acid. And Snyder agar is also differential because it has a lot of glucose in it and a pH indicator. So we're looking for glucose fermentation. And the pH indicator is bromcreosol green or bromocreosol green. It turns yellow with acid. And so the idea is you swab your teeth and gums, you inoculate the Snyder agar, we incubate it. The more of the Streptococcus and Lactobacillus bacteria you have on your teeth, the faster the agar turns yellow. So we examine the tubes at 24 hours, and if it's yellow in 24 hours, that indicates a high risk for cavities because you have a lot of bacteria making acid. If it's yellow in 48 hours, you have a moderate risk. And if it's only slightly turning yellow or still green at 72 hours, you have a low risk. But the idea is just to look for those two groups of bacteria which will ferment to produce a lot of acid. So the more of those bacteria you have on your teeth, then the higher the risk. Now the agar also contains a casein, which is a milk protein, because both Streptococcus and Lactobacillus also love milk, and so they'll use that to help grow as well. So any questions on the Snyder agar? Would have been so fun to do your all all of your teeth, but we can't do that this semester. Now the particular Streptococcus, the species, is Streptococcus mutans. And so it's the primary cause of cavities, Streptococcus mutans. So in order to reduce the numbers of these bacteria, you should brush twice a day, floss twice a day, and use mouthwash twice a day. Of course, ideally, you should do this when you're kids, right, when you're young. But of course, most kids have a problem with brushing twice a day or flossing twice a day or using mouthwash twice a day. So that's when most of our damage occurs, when we're younger. Unfortunately, that's the case for almost everything, right? And so later in life, you'll have to deal with, replacing fillings or getting crowns or dental implants to deal with all your bad teeth. But it's hard to get kids to follow good oral hygiene. And hopefully you guys do now, right? So any questions on Snyder agar? So continuing with normal flora, we had throat and skin. So for throat, we use blood agar plates. So remember, the blood agar plate is just a TSA plate with 5% sheep's blood added to it. And so that allows us to differentiate the type of hemolysis or hemolysis, as well as to enrich the agar and allow fastidious microorganisms to grow. So a fastidious microorganism is one that's very particular about its nutrient requirements. So plain TSA may not have enough nutrients to allow those bacteria to grow, but you add blood and then the bacteria grow without any trouble. But we do use it to differentiate the type of hemolysis. And there are three types, alpha, beta, and gamma. Alpha hemolysis is the partial breakdown of the hemoglobin and actually turns the blood agar kind of a greenish brownish color. So we see the alpha hemolysis here and here. In reality, all we ever see is the brownish color and not really green, but technically it's a greenish or greenish brownish color. And everyone has alpha hemolysis. Beta hemolysis is the complete destruction of the hemoglobin. And so the agar actually turns clear, and it's clear enough to see through. And we can see a nice clear zone around these bacteria, and all this is clear in here. And so many common bacteria are actually beta hemolytic, things like Staph aureus or Bacillus cereus, and of course Streptococcus is. But whenever students do this, it's possible to get Streptococcus mutans right from your teeth, and so that will show beta hemolysis, or the Staph aureus if you like to eat with your hands and fingers a lot, and sometimes Bacillus cereus. Gamma hemolysis is no hemolysis at all, and so the blood agar is not changed. It's still nice and red underneath the growth. Any questions on hemolysis and blood agar? So then we did skin and staph identification. So the idea was to see what type of staphylococcus you have growing on your skin. And we were using the traditional 3-species classification, Staph aureus, epidermidis, or saprophyticus. And so you would have taken swabs and swabbed part of your skin and then inoculated an MSA plate. We already know that we can differentiate Staph aureus from the other staphs because Staph aureus ferments mannitol and turns the agar yellow. So the MSA plate was the first test. Then you would take some bacteria from the MSA plate and inoculate a coagulase broth tube and then do a novobiocin antibiotic test. And so the coagulase and novobiocin could help to differentiate the other 2 species of staph. But coagulase is an enzyme that cleaves or splits fibrinogen to form fibrin, and those fibrin proteins associate to form a clot. And so the broth gets very thick and sticky. So we see a clot here, and the Staph aureus is positive for coagulase. Epidermidis and saprophyticus are negative for coagulase, and so the broth is still liquid. For the novobiocin test, you would have spread the bacteria all over the agar plate, applied the novobiocin disc, incubate, and then observe for a zone of inhibition. A large zone of inhibition indicates the organism is susceptible. A very small or no zone indicates the organism was resistant. Staph saprophyticus is resistant to novobiocin. Now, the most common type of staph that people have on their skin is epidermidis. About 1/3 of people have aureus, and just a small percentage have saprophyticus. But you should be able to identify the type of staph using these 3 test results. And so you need to know the results of this table. Remember, we said it's actually easy to remember because you only need to remember 3 out of the 9 results in that table. You need to remember that Staph aureus is positive for mannitol fermentation. Staph aureus is positive for coagulase, and saprophyticus is resistant to novobiocin. So all the other test results are the opposite of those 3. So if you know those 3, staph ferments mannitol, staph forms a clot, or a Staph aureus ferments mannitol, Staph aureus forms a clot, and the saprophyticus is resistant to novobiocin, then you know everything else in that table. So any questions on the staph identification or the throat cultures? So we also looked at water quality. Remember, for the water quality analysis, we were just looking for bacteria, and in particular coliforms. And coliforms are fecal bacteria like E. coli and Enterobacter aerogenes. And so we're worried about fecal contamination of the water because there may be pathogens associated with feces like Salmonella or Shigella. But those pathogens are not going to be present all the time. But if fecal contamination is a constant problem, you will find coliforms. So we use the coliforms as indicators of water quality. And so the standard for coliforms is no more than 1 coliform per 100 milliliters of water. and no more than 100 other types of bacteria per 1 milliliter of water. If your water is below those two standards, then it is potable, it's drinkable. But if you have more than one coliform or more than 100 other kinds of bacteria per 1 milliliter, then your water is not potable, right? You really don't want to drink it. So we use a membrane filter apparatus in order to filter our water samples, and we use water from one of the aquariums in the department as well as the pond outside. And so remember, the actual filter membrane will trap the bacteria. Everything else goes through the membrane. And then we apply a nutrient medium to the membrane filter. And so that nutrient medium this time was M. coli blue. And so M. coli blue turns E. coli colonies blue and other coliforms dark red. Any other colors we don't care about because they're not coliforms. So on this plate here, we see there's at least 5 or 6 blue colonies. So we know there's E. coli there. And the other colonies are dark red, so those are other coliforms. So clearly we have exceeded, right, the minimum standard for water quality, and our water was not potable. Now, there are some problems, and so there are some disadvantages of using a membrane filter as well as advantages. Right? The advantage is it's fast. Within 24 hours, you get colonies that you can see. It's quantitative. You can count those colonies and determine a concentration of coliforms per 100 milliliters. And it's sensitive because you can filter large volumes of water and find just a few cells. So it's fast, it's accurate, it's sensitive. But if your water sample is really turbid or really muddy, that will clog the filter. And if your water sample is highly contaminated, you would have to perform serial dilutions and repeat the process in order to obtain isolated colonies so you can see them and count them. So any questions on water quality? No questions on water quality? So we save the best for last, right? The sequencing. Remember, some of you haven't turned in the bioinformatics homework yet, so make sure and get that in by the end of the day today, right? So for the sequencing, remember that we had a bacteria and we had to boil them to extract the DNA. And then we centrifuge them at high speed so that all the cellular junk would come down to the bottom and the DNA would be floating in the supernatant liquid. And then we performed a PCR reaction. And remember the 4 ingredients of PCR. We have our template DNA, which we made. We have our thermostable DNA polymerase that came from a thermophile. We have all the nucleotides, ATGC, and we have primers. And remember, it's the primers that allow for the specificity of the amplification because the primers only bind to the gene of interest. And because that's the only place where it's double-stranded, that's where DNA polymerase will hop on and start replicating. So you set up your PCR reaction and then verify if you actually correctly amplified the right piece. We ran an agarose gel. So we were looking to see if you amplified a piece of the ribosomal RNA gene approximately 800 base pairs long, right? So technically 779, but you would have used the DNA ladder, and hopefully you would have seen that you had a band of DNA corresponding to approximately 800 base pairs, and that meant that you did amplify the correct piece. So if you did that, let's go through the PCR process again, just for review. So we have to heat up our sample to 95 degrees Celsius to denature the DNA and separate the two strands. Then we cool it down to about 58 and allow the RNA primers to find the correct complementary sequence so they can bind. Then you heat it back up to around 72 to make it warm enough to allow our thermophilic DNA polymerase to function, but not so hot that we separate the primers from the DNA. And then we repeat that process over and over again, 30, 40, 50 times. And if you started out with just like one piece of a double-stranded DNA, at the end of 30 cycles, you would actually have created over 1 billion pieces. So a PCR is a very powerful technique. It's very efficient and economical. So you sent out your sequence, or you sent out your DNA for sequencing, and it was sequenced using the Sanger dideoxy technique, which uses the same 4 ingredients as PCR plus an extra ingredient, and that extra ingredient is a dideoxynucleotide. So remember, dideoxynucleotides do not have the 3' or the terminal hydroxyl group, and so no more nucleotides can be added on after it. So when DNA polymerase randomly incorporates a dideoxynucleotide, elongation terminates. No more nucleotides are added after. So you set up 4 reaction mixtures, each with a different dideoxynucleotide. Every possible size fragment terminating with that dideoxynucleotide will be produced in that reaction mixture. So we have to separate those fragments using a polyacrylamide gel. Right? Polyacrylamide is very sensitive. It can separate DNA fragments that differ by 1 base pair. And so we load all the different reaction mixtures, and we turn on the power. It separates the fragments, which differ by only 1 nucleotide, and then we read the gel from bottom to top, because each band corresponds to a fragment that terminates with that dideoxynucleotide. So that's how we read the sequence from bottom to top. And so once again, C, T, I can't see this, hold on a second. It's not letting me, okay, here we go. C, T, G, C, T, T, A, G, A. So each band in the gel corresponds to the fragments that are shown here. Each fragment terminates with a specific dideoxynucleotide. So that's how we read the sequence from bottom to top. So questions?
So C is really the first one,
Yep.
even though it comes last.
Yep, because it's the smallest fragment. So it comes first. Remember, we're reading the complementary sequence from bottom to top. So
Oh, right, it's a complementary sequence. Got it.
any other questions? Okay, so remember, your lab exam starts at 1. On Wednesday. In the lecture before, we'll have the review for the optional final lecture exam. So if you think you need or want to take it, then show up for lecture. And then later that day, I will try to have your cumulative final grades calculated, so you can see whether or not you need to take the optional final or if it can help you. So for many of you, or some of you, Wednesday will be your last day. So any other questions?
Okay.
Yep.
Is the percentage that's on the grades for Blackboard now the closest? Is that it? Is that exactly what it is? Our grade?
Well, I haven't finished grading all the lecture exams yet, but that will include your lecture exam grades. The only thing left will be your lab exam, or the bioinformatics homework if you haven't turned it in yet, right? So
Right.
the rest of you, make sure you get that in.
So we can decide on Wednesday if we're gonna take the final. Like it's okay if we decide then, right?
Oh yeah. Mm-hmm.
Okay. And then if we don't take it, we just don't do anything, right?
Well, hopefully you'll send me a nice email saying goodbye.
Right. Yeah. Okay.
All right.
Sounds good.
All right. So any other questions from anybody? All right. The rest of your exams will be finished very, very soon. So I can check those out and see. And then I'll talk to you guys or see you guys on Wednesday. Okay. Okay, everyone. Remember, don't forget to give me the bioinformatics.
Thank you, Professor. It was a great semester.